Viability. Now, seeds that germinate and develop, we say that those seeds are viable. The viability can be expressed as a percentage. So viability can be defined as the total number of seeds that are expected to germinate out of 100. So you can test 100 seeds from a packet of seed that you purchase commercially, and you have to determine how much of those seeds germinate. So if 90 out of the 100 seeds germinate, well, then you can say the viability of that packet of seed is 90%. Cool storage will maintain viability for a longer period of time, meaning if you store the seeds in a cooler condition at a lower temperature, the seeds will remain viable for a longer period of time, and that is why some persons will store their seed in a refrigerator. So the viability test is most often done on seeds that a farmer may have for a long period of time. So the viability test is mostly done on old seeds. Saccharification. The diagram here is showing how saccharification is carried out. Saccharification involves the weakening, the opening, or altering the coat of a seed to encourage germination. So this practice on seeds that have a very hard outer coating, and by damaging the outer coating a little bit, you're allowing the seed to germinate a little faster. Next, we're looking at the way in which seedlings can be grown. So we have two main systems. We have the container system and we have seedbed system. So using container system, we use potting soil. The potting soils are commercially available, meaning you can purchase these potting soil, or they can be made locally. And if you're going to make potting soil locally, this is a recommendation that you have 3 parts of clay, 3 parts of pen manure, 1 part of sand, and you add 1 part of sawdust. Or instead of sawdust, which is more a coconut fiber might be more easily accessible, or bagasse. In addition to that, limestone may be added to the potting soil that you're making. The soil should be treated. Either you treat the soil using steam, using heat, or you treat it using methyl bromide. And the treatment here is to get rid of microorganisms, to get rid of bacteria, to get rid of fungus that will cause diseases in the plant as they germinate. The ideal pH to achieve on the soil would be around 6.5, will be around 6 to 6.5. And one of the reasons for adding the limestone is to adjust the pH so that you can have the ideal pH to grow the seedlings. So Seedbox. Seedbox are used for sowing seeds, for thinning out seedlings, and then to raise a seedling to a certain stage before you transplant them. There's a recommended size that you can use to make your seed box, and the recommended size is that the length should be 35 centimeter, the width 25 centimeter, and the height 7 centimeter, and that is shown in the diagram on the right. At the bottom should have small slits, small holes, and these holes are to allow the water to drain easily. Because if too much of water remains in the soil, the seedling will develop all sorts of diseases. Fungus will start to grow in the soil and it will affect the plants. The slits or the openings or the holes that you have at the bottom, they need to be covered. They can be covered with grass or straw, and that should be enough to prevent the soil from being lost. The seed box is then filled with potting soil, and then you level the seed box. Seedling trays can also be used, and seedling trays are now mostly used instead of seed box. They have compartments. Some may have around 100, some may have much more than 100 compartments, depending on the size of each of the compartment. And 1 seed is usually grown in each compartment. Because the seedlings are in separate compartments, thinning out will not be required. Thinning out is when you remove seedlings that are too close to one another. Thinning out allows you to space the seedlings far apart from each other so that they do not have to compete for light, for nutrients, and other resources. When the seedlings are too close together, well then both of them may not grow, or if you have several of them close together, they will not grow as healthy as if you have one single seedling in a single compartment. The seedling tray system allows easy transplanting. When you remove the seedling, it's easy to remove the seedling and transplant it onto the field. So the advantages of container system: the seedlings are cared for in a nursery. Because you're using a container, you can move it into a nursery. A nursery would be an enclosed, it could be an enclosed area or an area that has a shelter to prevent too much of sun or too much of rain from getting onto the seedlings. The containers can be reused, whether they are made from wood or from plastic material, they can be reused. And one of the good things to do is that before they are reused, the container should be washed and disinfected. You can simply add bleach into water and wash the containers before you reuse them. Each seedling can be transplanted with a ball of soil around the root. So when you remove the seedling, a ball of soil is also removed with it, and that's a better way of transplanting. If you're going to transplant and you just remove the plant and you only have the root, well then it is not so easy for the plant to survive after you transplant it. Seedling can be well rooted into the potting medium that you're using, and those are the advantages. Next we have the seedbed system. So seedbeds are used for a large number of seedlings or large number of plants. For example, rice farmers, they will place the seed directly onto their seedbeds. Or if you're cultivating cabbage, well then the seed can be planted directly onto the seedbeds. The seedbeds are close to the field plot. The field plot is where you're going to grow the plant after it matures. Typical size of a seedbed would be 1 meter in width and 3 meters in length. And the seedbed should have a cambered surface, meaning that it should have a little curve to allow the water to drain very easily, so that in rainy season you do not have too much of water accumulating on the bed. Sowing seeds, well, there are different ways in which sowing of seed can occur. Broadcast by hand, or you can just scatter the seed evenly throughout in a seed box or in a seedbed, and then you cover the seed with a thin layer of soil. The seeds can be sown in shallow drills made with a dibber, and the dibber is shown in the diagram there. So the dibber is used to create a small hole, and then you place the seedling in that small hole and you cover the seedling with a thin layer of soil. If a seed tray is used, 1 seed per compartment, and we looked at that before. Tiny seeds, very small seeds, for example celery seeds, you can mix them with sand and then apply it on the soil, or you can even you can add them to water and then apply the water onto the bed. Plant nursery. So a plant nursery is where the seedlings are grown before they are transplanted onto the field. And the main purpose of the plant nursery is to protect the seedlings from the harsh environmental conditions, protect the seedlings from direct sunlight, it should also try to protect the seedlings from diseases. It's easier to manage the seedlings in a plant nursery rather than to grow the plant directly on the beds in a field. Some of the features of a plant nursery would be that the structure should be made of something that is strong. It should be a wooden structure or a steel structure, a concrete floor, and the roof with transparent polythene material. That's like a plastic. And the roofing, or the roof that is used, is really to protect excessive sun from getting onto the plant and excess rain. So that brings us to the end of this section where we looked at the propagation of plants. So if you have any questions, once again, do not hesitate to ask those questions. Until the next lesson, I wish you all the best.